Hey everybody, Dr. Rob Silverman here. How are you? I'm excited to be here. I got a superstar guest today. We're doing guest week, and guess who we have? Dr. Erika Gray. She is the smartest person I know in the room. She's going to talk all about some exciting topics like genetics, genomics, why you would want to know your genetics, your genomics, the difference, and things like that. So many of you have written into me and said that I don't understand what 23andMe is. Why do they call it 23andMe? Should I take it? What the outcomes are. We've got the co-founder of Toolbox Genomics right here, right now, to talk about all those things. Hi, Dr. Gray. How are you?
Doing great. Thanks so much for having me, Rob.
Oh, it's my pleasure. It's exciting. So why don't you tell everybody real quick, why the heck, forget your bio, we're going to get to it. Why do I want to take a genetic test? Why do I want to know my genomics?
So I think the best way to describe it is when I was a little girl, my father's a civil engineer, and he would take me to the city, and that was the first place we had to go. And it was the most boring part of the entire trip. And we would go there to get the blueprints. And so one day I said, can we just skip this part because this is really tedious? Let's get to the fun stuff. And he said, no, this is really, really important because if without the plans, I don't know where to put the building. I don't know where the property corners are. And that's what our DNA is. That's why it is so important for us to get that inner blueprint of who we are. And when we take a genetic test, that's like going to our personal city and saying, what is the lay of the land? What do things look like? And it gives you so much insightful information about how do we handle nutrients? How do we detoxify medications? How do we detoxify for the environment? And how do we work with different, like our family history? Where's it coming from? And so genetics is really just like that inside look into who we are.
Outstanding. That's exciting. Because you know what? I used to love the idea of like, I wonder why I have hazel green eyes.
Yeah.
Right? You know, my sister has blue eyes. My dad has blue eyes. My mom has brown eyes. I have hazel eyes, and my sister has blue eyes. And I know that blue is recessive. Is it something like that? Is that idea of why we express something in the genes and what we can express?
Exactly. And so, for example, like your sister has the coding for blue eyes and we can see the blue eyes, and for you, it's hazel eyes. But when we get to the topic of health, it's more subtle than that. So it's the same thing. We have the coding, but maybe we don't see it immediately. Maybe we don't see that rising glucose level, or we don't see the brain changes that we're automatically born with, depending on what our genotype is. It's a more subtle thing, but we can measure it and we can really get some fundamental information around that, so we can make good lifestyle choices for ourselves.
So what you're saying is I can't change my eye color, but I can change my health expression.
That's right. So just like you could change your eye color by putting on a different color contact, right? But at the fundamental level, you still have hazel eyes. So it's the same thing for us. At a fundamental level, we may have a genetic predisposition to diabetes, but we can put things into our body that's going to change the expression of it.
Outstanding. So a lot of people ask me, what's the difference between genes, genomics? Don't worry, and don't worry, Dr. Gray is going to excite you. This is not, it's not, she's not going to do her PhD pontification now. She's going to let you know it's genes, genomics, and what's epigenetics.
Okay, maybe we should start at the top. So let's start with genetics, right?
Mm-hmm.
So we've got our chromosomes, and we know that. So that's like if you go into your kitchen, you've got these big pieces of appliances. You've got a dishwasher, you have an oven. Well, what if something happens and the builder forgets to put a dishwasher into your kitchen? Well, that's kind of a problem for some people, or they forget to put an oven in. So when we talk about genetic changes, we're talking about it at a chromosome level. So Rob had mentioned 23andMe, we get 23 chromosomes from mom, 23 from dad. That gives us a total of 46. And within that, now we can get into the DNA. So within the chromosomes are the tiniest little measurement of it, and that's DNA. And then from there, now we get to look at genomics. So now you're in your kitchen and you have a recipe, and you're going to make some changes within that recipe, and it's going to change your bread from being salty to sweet. Again, it's still bread, but how it tastes is going to be different. So those are the high level. And then we say, well, what if I were to pair that bread with some fruit juice? Well, now the whole change the flavor of it's going to change in your mouth. That's epigenetics. We already know what we're expressing. The way it interacts with our environment is going to change. Sorry, it's going to change. What ends up happening with our genes, how we express it, is really being pushed by the environment. And so again, as we mentioned earlier, your genes never change, but the expression of it does. And that's where the environment is pushing.
So we've heard the expression, we use it all the time, genes are not my destiny.
That's
correct?
right.
So if somebody walks in my office and wants to do a body contour, they want to lose weight, and they say to me, mom's heavy, dad's heavy, my brother's heavy, I'm destined to be heavy. Is that correct?
Not necessarily. So they may have, so there's two things, right? One of them is they probably have some genetic predispositions around obesity, snacking, maybe even fat or protein metabolism. But if you know that information, you can make much more educated decisions around your lifestyle choices. And the earlier you know this information, the sooner you can take action on it as well. And I was just going to pop that. Yeah.
Yeah, you want to do that again. You froze for a split second, and I know that was unique and needed.
Yeah, absolutely. So mom is heavy, dad is heavy. Well, you may have the genetic predisposition for obesity or snacking, something which is going to destine you on that trajectory for weight gain. But if you know this information early and you take action on it, you can essentially keep those genes from expressing themselves in that obese fashion.
So kind of like a personal story. I'm not heavy. My sister and I have the same genetic outcome. She is unfortunately heavy, not picking on my older sister. So, but we have different lifestyles. We eat different foods. We have different exercise regimens. So I'm not letting that gene be expressed where she may have let that gene express. So Maymay just wrote, genes are not your destiny.
That's right.
That's right.
And I think a really great highlight of what you were just saying is when the twin astronauts one of them went to space and the other one came back and the other one stayed here on Earth, and people said, oh my gosh, their DNA changed. No, it didn't. Their DNA did not fundamentally change. The expression of it did. And it was amazing because you could actually see physical differences between them because of what the pressure, the environmental pressure of space and what it did with the DNA versus the one who stayed here on Earth.
That's a great example. He was 2 inches bigger. He was heavier. It looked like Big Brother where they started, because one was on a space station. The other one was down here. Their genes were expressed differently because their environment changed.
That's right.
And an environment isn't just where you live. It's the supplements you take. It's the food that you eat.
It's your sleep. It's microbiome. All of those are when they work in concert with your genes, that's your epigenetics.
Excellent. Excellent. So people know. So for me as a practitioner, I love to take these genetic tests right here. And it actually says optimize your genetic potential. I kind of like that. So that's the Toolbox kit. We're going to keep referring to this and a few others. We're going to highlight some. But I just want to ask one thing that's really important. Everybody's heard of Alzheimer's. Everybody's scared about having Alzheimer's. And there's a gene, APOE or APOE4. Why don't you talk about that? Let's get that going, a little methylation, and then let's get into the kit so everybody knows what you're looking at and traits and snips and stuff like that.
Yeah, absolutely. So before we touch there, one of the things we didn't talk about is a single nucleotide polymorphism, because that's really where APOE gets to. And so it is a change in the base pair of your DNA. So we have an A, a C, a T, and a G. And how they arrange themselves is going to dictate a different protein expression, how things bind at the receptor, or even how we convert an inactive form of our nutrient to an active form. So for example, if we just take this sentence that I quickly wrote up. So a man saw a dog. Okay, so he saw a dog. Maybe he's going to call the Humane Society. He's going to take action to help save this dog. Well, what if instead of seeing a dog, he saw a dot? So one letter change, very different outcomes of what this man is going to do. And that is a single nucleotide polymorphism, or as we affectionately call them, SNPs. So when we are talking about APOE is a gene that codes for some cholesterol transport. And at a fundamental level for people in their brain, the way that their brain develops in utero is actually different from what we call a 3-3. So when we're looking at these differences, it is a single nucleotide polymorphism. The average person is a 3-3, which means they have an average risk for Alzheimer's. But then if you get that base pair change, now you're actually considered a 4. And the 4s have a whole unique changes in their brain. They are at a higher risk for worse outcomes from concussion, from TBI. Their nutrient status for their brains is different. So it's really interesting that this one little base pair change can really have such a big effect on our lives. And we get to take action and really help support that as well.
So you're checking on these kits for snips. You told everybody what a gene was and traits.
Right. So if you think about it, you have a trait, which could be a nutrient deficiency. It could be a disease, something you have an ICD-10 code for. It could be an enzyme speed, like our CYP enzymes. So we look at, and we call those traits. From there, we've got genes that go into those traits. And then the next level down is SNPs, the single nucleotide polymorphisms. And so we check for all of those. And then we tell you, what is your result? Are you a plus for the variant, which means that you're homozygous? Are you heterozygous, plus minus? Are you do you not have the variant where you're negative? And so be a minus. And so that's how you can look at it. And then that's how you're going to be able to help your patients really understand this is why this is going on in your body. This is why your family history is playing out the way it is. And now this is what we're going to do to take action around it.
Absolutely. So obviously that's going to catch people's attentions. Alzheimer's is at the front of everybody's consciousness. So you know if you have a genetic disposition. But again, since genes aren't your destiny, it means you can avoid it. So I have a 3-3. So that means life is really good. But if I decided to live up by a highway, eat fried foods, have some sugar sodas and things like that, I may change my genetic outcome and I could become susceptible. So for me in the office, I love to use these different kits as baseline for outcome. So can we go through one of these kits? So why don't we do one like, let's start the easy one. Why don't we talk about weight loss and nutrient optimization?
Yes. So nutrient optimization is my favorite one because everyone wants to know, should I be taking these supplements? Am I sensitive to gluten? Am I sensitive to lactose, et cetera? And that is really where you want to start because now you're going to look to see, do you have a genetic predisposition around folate, around B12, choline, magnesium? Because you as a practitioner, the way you're going to dose your patient, the way you're going to follow up with further testing with your patient is going to be different if they have a genetic predisposition. If you think about it's like that magnifying glass, keep an eye on this. This is something you want to track. And then especially when you compound it with either family history or lab work, or even a physical assessment that you've already done on this patient, this is something that you're going to help work with them on. It's going to be a more tailored and personalized protocol. And another one, for weight loss, there's the FTO gene, which is affectionately called the fatso gene. And it was designed to get us through famine. So it's genius, right? We would, if food was available, we would stock up and we put on the pounds. Well, a lot of people have that variant. And so what it means is that you have like a 3 to 5 pound on average weight gain over time. And so now for a lot of people, they will compound that with the processed foods, with your omega-6, omega-3 imbalances, as you mentioned, the sodas and the sugar, et cetera. So knowing that information, now you can explain to your patient, there's a genetic predisposition for the weight gain. We need to take a much more targeted, focused protocol for this. Maybe we need to do intermittent fasting. We've got to stop eating at 6 p.m. because we know we have this gene and it's being expressed. So we need to work to undo that, so to speak.
That's fabulous. I want to stick with that. I just don't want everybody to think like I'm not listening to Dr. Gray. What happened is my screen is just her and I. So I'm actually communicating the phone. So if you see me look down, so what I want everybody to do is, of course, I want you to like it, comment, ask a question because I'm getting some DMs, but feel free to ask a question on the thread and of course share it. So question number one was, it's a really good question. So you were talking about all these different genes and traits. What's your probability? And I think they're meaning what's your correctness, your sensitivity, specificity, what's your correctness? It's a good question.
So yeah, it's a great question. So we actually set everything at industry standards. So when it's a 95% or greater, what they call rate. And so that's the accuracy. But we also went through an additional step of clinical validation, which then raises us up another level. So that's at 98% or greater. And those are the industry standards that everyone follows.
That's great. I got another question for you. How are you different than 23andMe? These are DM questions. They're not putting them on my thread.
So a couple different ways. So for 23andMe, they actually, in their latest array, they pulled off a bunch of SNPs that a lot of clinicians were interested in. So our array has a 30% difference from 23andMe. Plus, we go through and we do all of the interpretation and the collating of the different SNPs into different panels for you, so you don't have to do it. Because we do everything from the kit all the way through to the interpretation, we can control and protect privacy. You're not taking your raw data and sending it out to a third party. So we have complete control there. And we also allow customization. So you can go through and pick and choose different traits that you would like to put together for your patient. After you have swabbed with us and we've done the sequencing, because it's just a quick cheek swab, it's really easy, then you can actually unlock other panels at any time to give your patient more information and make it more cost effective.
Well, you got a shout out. You got a shout out from Wisconsin.
Aww.
And then you got a shout out from a doc who says great information. And I got another DM that said, so wait a minute. So you can, with these kits, I'm extrapolating what they wrote, you can do a baseline for weight loss. So tell me how the kit and what your gene probability is will help that person lose weight. That's essentially what they're asking. So for instance, for me, I found out that I don't do well with saturated fats.
Right. So, okay. So first thing you're going to do is you're going to swab your patient. You're going to send that information into the lab, and then the data will be sent back to us, and then we will put together that report for you. In there, we're looking at a couple different things. We're looking at saturated fat metabolism. We're looking at your ability to lose weight with a higher protein diet. We're looking at your propensity for snacking. We're looking at yo-yo dieting. We're also looking at stress response, because we know that a lot of people are stress eaters. So when you understand how your patient perceives the world, is it a scarier place, or are they looking for a fast dopamine rush, possible addictive behaviors? Now you're going to be able to give them even more focused information, but from a psychological perspective as well, that's going to resonate with them to get them to be more compliant and then follow through with your protocols.
Now, I asked her to keep it real simple, and she's doing a great job. People don't understand how complex it is. So I think we're getting some practitioners on. So let's talk about something a little bit more complex.
Okay.
And the thing that they all struggle with, even though we talk about it. Let's talk about methylation. What is it? Hyper, the kind that you have to, and what it means. It means if you can't methylate, what's the big problem is.
Okay. So let's start with the queen with bees. I think this is the best way to highlight what methylation is. So everybody starts as a worker bee, but then at one point, the worker bees select a particular individual and they feed that one royal jelly. And that one now has turned into a queen. So that is what methylation is. And what ends up happening, so it's essentially, let me rephrase that. That is an example of what methylation is like. So it's one carbon metabolism. It's just the movement of this carbon with three hydrogens onto different molecules. Depending on where we are in our life, we want more methyl groups on our DNA and we want less methyl groups. So it's really important to understand where people are in their lifetime and what is happening for them. So what ends up happening is that these methyl groups go onto the DNA, and it's like they're little blockers. And so when the DNA comes along, polymerase, I believe, comes along and copies it, it's going to skip over sections of the DNA. And so the thought behind a lot of diseases like cancer is it's hypomethylation. We don't have enough methyl groups, and then as a result, this wonky pieces of DNA are getting copied and then propagated through in other cells. But it's important to know that in certain times of life, like during pregnancy, we might have an increased need for methylation. So we might be hypermethylating there. So it's this balance of where we are in our life, and we need adequate methyl group availability. And our methyl group availability is primarily generated through S-adenosylmethionine, SAMe, which originates from folate. And so folate is considered one of these master methylators because of what it does in pathways further downstream.
Excellent.
Is that simple enough?
Yeah, that was good. We had to give him a little technicality. Somebody, I just put it in the link, Doc, Melanie. It's Toolbox Genomics code, lowercase everything for right now, Dr. Rob. That's going to allow you to get a discount. So if they're asking about the lab, why don't you tell them some of the kits? It's Toolbox Genomics. Why don't you tell them about some of the kits that you offer and what it does? Now, just remember, everybody, it's a cheek swab. So some people don't like serum for some reason, they don't. And a couple of people don't like the finger stick, the cheek swab. No one's ever said no to a cheek swab.
And you can do it on your young children and your elderly parents as well, because a lot of times they don't have enough saliva production to spit into a tube. So one kit, we've got, I believe it's 15 different panels that you can then choose from. So what I recommend everyone does is either you start with nutrition optimization and then you pair it with something like Detox. You can pair it with Dr. Rob's panel, like the gut brain panel, or TBI concussion if you've got some young athletes that you're concerned about. And start there. Just start with 1 or 2 panels for your patient. And then if you're really concerned, or you know that there's a strong family history of something, we're just going to take Alzheimer's, for example, you can add on the Alzheimer's trait out to that particular panel for an additional $10. So it makes it really accessible. Once you get those results back, now you're going to assess your patient. You're going to figure out what's going to be the plan, how you want to move forward. And we offer free consultations at any point. So just you can make an appointment with any one of us, and we'll help you either pick a panel or interpret the panel with for your patient's results. And then now you can go through and add on additional panels as well at a later date or just at the next appointment with your patient.
That's great. All right, so I got a couple of questions here. How long does it take to get the panel back? Because 23andMe took 3 to 6 weeks.
Yeah. So it is between 2 and 4 weeks, and we're running right around 3 weeks currently.
And that's correct, because genetic tests take a little longer. If I take the blood, it usually takes a month. That is standard, quick, if you will.
Exactly.
Can a individual get it? I'm not a doc. Can I get some kits?
So if you're an individual with Dr. Rob, we've got a special affiliate code set up, and you have access to certain panels. So you have to go to toolboxgenomics.com/Dr. Rob, and that's DRROB. And then that'll give you access to nutrition optimization, the weight loss. We have a fitness one. And then we also rolled out a new one called respiratory resilience to really get a deeper dive into some of the cofactors, which may be playing role in supporting our immune system or viral predisposition as well.
Yeah, that's critical. So that one is for the COVID-19. So a lot of people, somebody just said COVID-19. So as you know, we all do the antibody test. So we've got the viral, oh my God, help me out.
The nasal pharyngeal swab.
Yeah, the swab. Thanks a lot. I've been seeing patients and wearing, stuff and losing my marbles a little bit, and I'm due to eat. So I apologize. So we've got the swab that's going to say yes or no. We've got the antibodies to see if you've been infected and you have your natural cascade. What I like about your respiratory is not everybody's been exposed. I mean, less than 20% of Americans have the antibodies. We know that the virus hasn't gone away. So how do I know if I'm susceptible, and what do I do in that susceptibility? So I think that you may want to expand on that and let everybody know that what you're really testing for and what's going on, because you have the interleukins in there, you have the TNF, etc.
Exactly. And so what we did was we looked at the literature and we found that there are very clear comorbidities or cofactors which accelerated someone's either development of the virus or in how serious it became. So the areas were blood pressure control, glucose regulation, dysregulation, really more specifically, inflammation, oxidative stress, and then nutrients. So vitamin D3 has just gotten a ton of publicity of late. Glutathione, selenium, zinc, all of these nutrients play a big role with our immune system and how the virus is going to replicate within our bodies, going to be more virulent or not, and how does our body mount an immune response as well. And so, as Rob was saying, inflammation is really important. Those interleukin 6, TNF alpha, it's essentially you have a more hypervigilant immune system. And so you have a pathogen that comes in, and now instead of it saying, oh, here, let's bring a few bacteria in and let's get this under control. It's like, holy cow, we need to mobilize all armies and let's get going on this. And so it's like an exaggerated response on this. And we also know that vitamin C plays a big role in dampening cytokine storms. So that mobilizing of all the armies is really that cytokine storm that we've seen with a lot of patients as well.
So you're testing for propensity towards specific interleukins, the ones that lead us down the cytokine storm, interleukin 1, interleukin 6, etc.
So we're doing 6, 10, 23, and then the TNF alphas.
TNF alpha. And
Yep.
You're also testing for can you make glutathione? Can you make a good vitamin C? Will you absorb it? Stuff like that.
Can you transport it? Is it getting to the cell? Because that's the other part of it, right? We can take it in, but if it's not getting to the cell, sometimes we need to take even more to really push it and shove it into the cell, so to speak.
That's fabulous. So that's the respiratory one. You also have a detox one that we never talk about much, but detox may be right. It may be real important because detox feeds the gut. We all know you have to take care of your gut in your detox, and detox may help you because there was a study that came out that air pollution increased your incidence of COVID infectability by about 15%. So you do have a detox one.
We do. And so we look at both phase 1 and phase 2 detox. So we look at the CYP enzymes in phase 1, and then we look at the next steps like the glucuronidation, the acetylation in phase 2. And because you need to know both. And so really when you start looking at that, and the way we've set it up is we do a modified polygenic risk score. So I believe it's page 4 of the report, you will see at a high level, is your patient at a high risk, medium risk, or low risk from a genetic perspective for having a difficulty in these different pathways. So we can look at UGT, which plays a role with bile and breaking down lipophilic substances. So a lot of our hormones go through that pathway. We can look at the CYP enzymes like the 1A1 or 1A2, which is going to play a role with estrogen metabolism and the hydroxylation. So those are going to be areas that we want to look at before we start making recommendations around maybe it's hormone replacement therapy. Do you need more DIM, or do you need to stick with actually go up to I3C, because you're going to see, okay, there's a particular issue in this pathway. And then we always want to do some follow-up testing to see if that particular issue is being expressed at that moment as well.
I got a DM question. This one's a great one. Okay. So they say I take the test. I find out that I have a genetic propensity towards something. I do a 6 to 12 month protocol. I'm completely compliant. I go back and I take the test. Did I change my genetics?
So your genetics never change. So you're going to do this test once, but it's going to be the phenotypic testing that you're going to be using as those guide or those markers for you, or even if it's just physical exam markers as well. That's what you're going to be using for seeing how that gene expression is changing in your body. There is some new technology that's coming out that we're super excited around, where it's going to actually look at methylation and with epigenetics, but that is in the pipeline later this year. So that will be a more specific investigation of that.
And we're definitely going to get you on for that. I think this person is stuck a little on this.
Okay.
He said, okay, so my genes are here. So obviously they're concerned about a couple of genes. They're doing everything they can not to express it, kind of like me trying not to get heavy.
Right.
What happens when they, great line, when they rethrow the dice if they have children?
Yeah.
Does that change?
So it can absolutely change with your children because it's going to depend on, so let's say you are homozygous for the variant, right? So you have two risk alleles. And if you are partnered with somebody, you have children with somebody who for the exact same SNP is negative, they don't have the variant. Well, your children are going to be heterozygous for it. So depending on what particular SNP that is and what it codes for, it may or may not be an issue. Now, let's pretend that you are heterozygous and your partner is also heterozygous. Well, you can have one child who's going to be homozygous for the risk, and you can have another child who's going to be homozygous who doesn't have the risk. So the real interest often comes up in people who are having children when they are heterozygotes in a particular SNP. So that is where, when you roll the dice with your children, you will see a different combination and potentially a different expression of it. So for example, like COMT, I'm an average metabolizer. I'm a heterozygous. My husband is homozygous for the slow variant. Our daughter is average, and our son is a slow metabolizer. And how they perceive the world is definitely different.
That's fascinating. So, people always ask that now you have a PhD, you're a PharmD.
PharmD, yeah.
Right. They want to know why genetics, why genes, like why would you get involved in that? What's your story?
So pharmacogenomics is something we learn about in pharmacy school because we know that it's been documented over time that the way people break down their medications is different based on ethnicity, gender, and then just fundamental differences within various ethnicities. So when my parents did their 23andMe and brought me their results, and then I found out my father was an APOE4 carrier, they said there was the classic question, so what do we do? And it just really made a lot of sense because it brought together all the training I had in pharmacy school, the knowledge with pharmacogenomics, but now I actually get to apply it to the area that I love, which is really what we call nutrigenomics now. It's that interaction of our nutrients with our genes, the environment with our genes, and what can we do about it? So that is how we ended up I ended up co-founding Toolbox Genomics because we wanted to give people actionable evidence-based recommendations around their genetics because it truly tells such a powerful story for them.
One more question. It's really good. So they're talking about the detox one. So drugs get broken down in the liver via detox. Wouldn't you want the doctor to maybe take this before they start prescribing a plethora of different drugs to see outcome?
Absolutely. So the one caveat I'm going to put on that is you also want to, if you are going to be starting polypharmacy or you have someone who comes in with polypharmacy, you do need to do a pharmacogenomic specific test. And there's just some additional level of scrutiny of regulation that goes into it because you're actually changing dosages for people. So we do not have a pharmacogenomics profile at this point, but those same enzymes that are in a pharmacogenomic profile are in our detox. And so at a high level, you can start assessing what pathways may be affected and where you want to take another look and potentially do a further pharmacogenomic test for that patient.
Now, a couple of people asked how they can get in touch. So I will add a link to Toolbox Genomics / Dr. Rob. You can get anything that you want. If you want to see more about Dr. Gray, it would be Toolbox. She's got a nice little picture there with her whole bio and everything like that. So you can do that. You can follow them at Toolbox Genomics on Facebook.
Yes, you can follow me personally at the Gene Whisperer and that's ER at the end, on Instagram as well. And then if there's any practitioners that really want to reach out to me, send us an email at info@toolboxgenomics.com, and then they'll make sure that the email gets to me. And one thing I wanted to mention is that if you're a practitioner and you're looking to utilize this in your practice, you want to go to toolboxgenomics.com, sign up there, and there you want to put in the referral code for Dr. Rob, because that is going to ensure that you get special pricing for practitioner and access to practitioner panels as well.
Outstanding. So that was great, right? It was easy. It was a lot of fun.
Absolutely. It's always great to chat with you.
Oh, and I love the expression of the snips. I can't tell you how many people come in and say, well, what's a snip? And it's just that change. And aren't there like 3 million snips?
Yes. Oh, it's huge. I mean, and most of them don't do anything. I mean, they play a role, but it's the small percentage that we're really focused on. And it's just so exciting to see the differences between all of us as individuals, and then tracing it back to the genetics and having an understanding. I think our brain just always craves the why, and that's what genetic testing supplies for us.
Again, I'm looking at the phone because I can't get it on my screen. So Dr. Betsy said that was great. She thanked you a lot. We've got a couple of other shout outs. Everybody said it was really interesting. I got a DM. Somebody said, I never understood genetics and genomics. It was always difficult. So people were afraid of it, but they do. Why did they name it 23andMe? That's another question they just asked. So I know we're giving them too much love, but we're not trying to give them love, but that's a standard. And
yeah, no, of course. So we were actually joking. It should have been called 46andMe, but it's because you get 23 chromosomes from each parent. And so
it's a pair.
Yeah, it's a pair. Yep.
That's why it's 46.
So that's right.
Great. Well, you know what? It's been great. I'm so happy you could come on. I learned something every time you come on. I realize I need to take more tests. So if anybody's interested, you can see I'd be very happy to show you my genetics. If you know me, you can match up what you think, because genes are not your destiny.
That's right.
This is Dr. Erika Gray, Dr. Rob Silverman, Proven Health Alternatives, always yours in health.